Gene editing systems have opened up a host of new ways to explore how life works, and also have the potential to help improve human health, produce better crops, and fight environmentally damaging pests. Here, 3 young scientists from the University of Otago's Department of Biochemistry tell us about how they plan to use CRISPR gene editing in their diverse research projects. We're not going to show you exactly how CRISPR works. There are many great explanations already available on the net. Instead, you will learn about some of the ways CRISPR is being used in research right now.
Well, my name is Soledad. You can call me Sol. Come from Buenos Aires. That's the capital of Argentina. Huge city. I just got a grant to start studying the circadian rhythms in legumes in a small little legume that calls Medicago truncatula.
Okay, so what's a legume?
A legume is any plant that makes a vine with the flower. So lentils are a legume, peas are legumes. They actually are really big source of protein for humans, and it's very used for livestock, for feeding livestock, especially alfalfa, for example.
Sol wants to know how legumes adapt their circadian clock to the environment they grow in. One day we'll be able to use this to make plants better adapted to the new environments that climate change will bring.
Yeah, we call it the model plant of the legumes. So we use it if you want to have seeds just sooner than that will take about 3 months for the plant to give you seeds. Just a small population that live in different parts of the world.
Right.
So you take samples of that plant, you have the seeds, you have the stalk of the plant, you also see this in sequence, so we have all the genomic, all the information, and I measure something called leaf movement. So plants move even though we don't see them. So when you film the plants during continuous light, you will still see them opening and closing their leaves, but they will have their own rhythm that is set by their internal clock, because the circadian clock controls a lot of molecular physiological processes, like flowering, like the moment they germinate, the expression of genes. There is a number of processes. What I'm specifically looking for is one change in a nucleotide in the whole DNA that can help me relate how the plant grows there and their genetics, particularly looking at the circadian clock.
So how are you using gene editing to study this?
So once you measure all this leaf movement compared to the genomic and try to find responsible genes for this difference that you see, you try to mimic these changes using CRISPR-Cas gene editing. The plant that has A and change it to C with gene editing, and see if changing this gene will make the plant behave like the other one that has a C. That's what I'm gonna do. I don't have a candidate gene yet because this project just started today, actually.
So you're testing the technique at the moment.
Exactly.
Make sure it works with
exactly. We put it in the plant using tissue culture techniques.
Sol and her assistant Shireen put the CRISPR editing machinery into new plants using a special kind of bacteria, agrobacteria, that transfers DNA into plant cells. They cut some Medicago leaves into pieces and mixed them with agrobacteria that have the CRISPR machinery encoded inside them.
And you incubate it with the bacteria is able to go inside because of the cuts that you make to the leaf. So it needs to be wounded for the agro to go inside. And that's leaving them with bacteria.
They then put the pieces onto nutrient-rich cell culture plates. Infected cells start forming calluses on the side of the leaf pieces.
So each of these squares that you see here are the explants that we took. That when they are in this media, they start developing calluses. Can you see the little white? That's embryos.
Oh, wow.
And then it's a little less humid. There, do you see that from the explant there is a little, you need to focus into there. There's a little plant.
Eventually, the calluses start growing tiny plants with the altered gene inside them.
So you get these plants that are ready to be moved to the soil because they have long roots.
Very cool.
So this was a leaf.
That was a leaf.
Once you try this and you actually see this, you can then translate it to other legumes like pea, lentil, or alfalfa, because there is a lot of genes that are common in both of them. This is a big family anyway, a family of legumes, but there is a lot of things in common. So you could find the gene that is non-homologous and try to see if when you change this gene.
I'm Gemma McLaughlin.
And what do you do?
I am a PhD student at the University of Otago. Invasive biology has always been my thing, and it's a big problem in New Zealand, as we all know. So it's like I actually want to do a PhD where I'm actively doing something to control an invasive species in New Zealand. And then it's like, well, wasps are everywhere, and everyone hates wasps. They came here accidentally. It was just hibernating queens and World War II plane parts was what they deduced is how we got them. But they've just done so well here that it's quite detrimental to other native species. Yeah, we had the worst in the world. We had the highest abundances in the world. And then CRISPR just blew up as well. So it was like, and we wrote a research proposal about CRISPR and RNA interference, and then fell into the lap of my supervisor, my future supervisor, Professor Peter Dearden, and then he just emailed me and said that's exactly what I want to do.
Gemma and her supervisor Peter are looking into how you can make a gene drive to eradicate invasive wasps from New Zealand. So you're getting rid of wasps
by genetic methods.
Genetic methods.
Yeah.
A gene drive consists of some bits of RNA that can alter or silence a specific gene, or insert a new one, plus the genes that encode the CRISPR-Cas machinery. The genetic modification uses CRISPR to insert and spread through a population at higher than normal rates of inheritance. Inside the cells of an organism that inherits one copy of the gene drive, the whole drive copies itself onto its partner chromosome, so that the genome now has two copies of the gene drive. In this way, the change is passed on to up to 100% of offspring, rather than around 50%.
And then, of course, there's the CRISPR method, which I think personally it's better. There's this new method of CRISPR called remote control, which it's like germline editing. So I can.
So what's a germline?
Oh, sorry, germline. Germline is like, the reproduction line and the germ cells, like the eggs.
So it's the part of the wasp that produces the eggs and the sperm for making the next generation.
Yes. So yes, they've done it in mosquitoes, so they just had to inject a female, and then the effects they wanted were expressed in all her offspring. Whereas opposed to normal, traditional, like 3 years old methods of CRISPR, where you're injecting each egg individually before they develop any more beyond a single cell. So if I did that, that would be really effective, because then I could just inject a queen and have all her offspring do what I want, as opposed to having a queen injecting every egg she lays within the small window before they develop too much as well.
So you're targeting, the cells that make the eggs rather than the eggs themselves.
Mm-hmm. So I'd like to target this gene called double sex, which is like sex determination in wasps. The eusocial breeding system with the haplodiploidy, where the haploid eggs, unfertilized eggs produce males and the fertilized eggs produce females.
To begin with, Gemma will visit a lab in the USA to learn the techniques they are using to introduce this system into bees.
What I'd like to do is the method called non-homologous end joining, which is pretty much you make a double-stranded break in the DNA, you cut out a section and then you join the ends back together. And for that, I want to target this exon on chromosome 3, and for the double sex gene that produces males. And so just cut that out so it produces a stop codon instead. So stop codon means that's the end of translation. We stop making proteins from here. So then it doesn't make males at all. So we get all females, and then we get a thing called like intersex offspring who can't reproduce. So eventually then you're only having females, they run out of males to reproduce with, and so they eventually die out. I wanted to essentially have them just make males because they're the ones that don't sting, but that's apparently too effective because then they don't reproduce the next generation and you'd just be killing one nest doing that. Whereas having females, because CRISPR works for multiple generations. So if we did that and had only females, they'd eventually run out of males to breed with and just hopefully live their life, die, and then not another generation is made.
There are many obstacles to overcome before a gene drive could be possible for wasps.
It's a bit harder with wasps because they're not a model organism and we don't have their genome online accessible. We're working on that now.
Oh, really? It hasn't been sequenced yet?
It has, just by some postdocs in the other room over there.
Oh, excellent. We need to understand their fertility genetics better and learn how to grow wasps in the lab.
It's a jam jar.
Yeah, I use these when I see queens at my house. I just put them in a jar and bring them into work. If you find wasps in September, that's a queen emerging. So I always get my lab mates to bring in any wasps they see. Yeah, and you can just store them in the fridge. Okay, so this is the first layer of a wasp nest. Look, this is the pistil here that they attach to sometimes trees. They're usually underground, but that's the first layer that the queen makes all on her own. If you flip it over, it's just little cells. They have really small cells at the start because she's just trying to produce as many workers as she can. And once she's got enough, then they take over and build the rest of the nest, and then she just becomes a baby factory.
And how long will they last in there alive?
These have been in here for weeks. They just go to sleep and they just sort of stop.
For months?
Yeah, they can. Well, the queens hibernate for months. Usually everything else just dies out. These guys I use for mating purposes. So we collect all the queens from a nest at the end of the season. The old queen makes like hundreds.
But most important of all, we need to look at all the benefits and hazards that a gene drive could bring and decide as a country whether this is a path we should take.
It does have multi-generational effects, and the biggest fears is if it would escape to another country. It's a valid concern. It's something that should be looked into. I mean, we shouldn't just release a whole bunch of genetically edited wasps into the wild, so we are going to look into things.
My name is Oluwatobi Iboda. I go by Toby for short. So originally I was actually born in Nigeria, but then my family moved to the United States when I was real young. So I did my undergraduate and my master's program at the University of Michigan in Ann Arbor. And I decided that I wanted to visit New Zealand for a year. I actually played rugby in the States for about 10 years, which is why I knew of New Zealand in the first place. I'm like, you guys have the best rugby in the world. I'm like, hands down. During the job search that I came across this program doing gene therapy, and I was like, it's already in New Zealand. Like, this is what I'm passionate about and have been passionate about.
So you've decided to do a PhD with Steph?
Yes.
Dr. Stephanie Hughes?
Yes. So I'm part of the neurodevelopmental and disease lab.
The neurons?
Yes, I would assume so. Now, my project within her lab is the first that looks at a gene that is associated with Parkinson's disease, actually. So with that, I'm studying this gene called ATP13A2.
What is it called again?
ATP13A2.
ATP
13A2. Or PARC9. PARC9 will be much simpler.
Some genes have terrible names. The PARC9 gene encodes a protein that's found in the part of a cell called a lysosome. Lysosomes are like the recycling center of the cell. Waste molecules are sent there to be broken down and reused.
When we have defects in this particular organelle, like especially in the neurons, like we see a lot of neuronal death, and it's been something that's been a feature of Batten disease, like something that we're noticing in Alzheimer's disease as well, and more recently Parkinson's disease. So that's what we are investigating. Yes, so there have been published research on this gene showing that mutations lead to the development of a juvenile form of Parkinson's disease. we see development around age 12 and deteriorating rapidly over the next few years. And it also presents with dementia, something that you're also not likely to see in children. What I will be doing actually is I'm trying to set up an induced pluripotent stem cell model of Parkinson's disease when ATP or PARC9 is mutated. So I'm using CRISPR-Cas in order to make my model.
First of all, Toby needs to grow some brain cells, neurons, in the lab. But you can't just take someone's neurons out of their head and start experimenting on those. So Toby has another way of getting neurons.
So I'm using these induced pluripotent stem cells to create my model.
What does that mean?
So these...
Where did they come from originally?
So these induced pluripotent stem cells were originally harvested from an individual. They were actually fibroblast-like cells, so skin. Now pluripotency means that it has the ability to differentiate or become several different types of cells. We harvest the fibroblasts, might treat them to turn them back into their pluripotent state, and we call those the induced pluripotent stem cells. And then through certain treatments, we're able to direct their differentiation process or their changing process into a particular cell type. In my case, neurons. These are my cells here. I'm growing these iPS cells on these 10 centimeter plates. These cells I will then be inducing into neurons later down the line. Now you can't see the cells here because they're actually quite small. But so we take them over to this microscope. There's not been a IPS or induced pluripotent stem cell model for ATP13A2, so this is real novel research here.
Cool.
Now the cells are actually looking quite good themselves. You can't see them yourselves, but let me just describe them for you. These are IPS cells, and they grow in isolated colonies. So these colonies might contain many different cells that are isolated and have distinct edges. So yeah, these are colonies of cells all the way over here. You usually want to harvest these cells and either begin the differentiation process, which I probably will be doing today.
Once Toby has made his neurons, he can use CRISPR to change the PARC9 gene in them and look at how it changes the way the cells look and behave.
We can make very specific changes to the DNA, taking advantage of the CRISPR-Cas system. There are several different versions of CRISPR-Cas that you would want to use just depending on your experimental parameters. When I'm producing this model, I use actually two different methods. There is the CRISPR-Cas, the normal system, which knocks out my gene. So I can introduce another copy that carries the mutations that we see in this gene that lead to Parkinson's disease. And then there's the CRISPR-I, which is a adapted system from CRISPR-Cas. So with my system, I use the crab effector protein, which turns off expression of the gene. So I'm able to knock down the gene without actually cutting or affecting the DNA underneath. Now these cells have not received treatment of any sort. So these are one of my control cells. So they're not transfused with anything. So the next step then is to begin to characterize my model. It would be good to see, all right, what sort of characteristics does this model have now? Are they similar to what we're seeing in Parkinson's disease patients? And if it is, therefore the model becomes more relevant and can be used for further experimentation in order to develop treatments in the future for Parkinson's disease.
Would you be thinking of trialing things like gene therapy?
Yes, so that will be down the line as well. Once we have confirmed the model, we've characterized the model, we'll try gene therapy to see if we can correct for it.
Are you still playing rugby?
Oh, not as much, Mike. You guys have been playing rugby out of the womb. I can't compete with that.